This is S2P3, Section 2, Part 3, the nervous system. This is lecture material for the second exam. There's a number of ways to look at the nervous system. Perhaps the easiest way to break it up is into pairs of things. So we can break up the nervous system into central and peripheral. The central nervous system we could break up into the brain and the spinal cord. Peripheral nervous system, we can break them into the afferents or sensory fibers and the efferents or motor fibers. The motor fibers we can break up into somatic, voluntary, and then the autonomic, involuntary. And the autonomic nervous system we can break up into sympathetic and parasympathetic. So looking at a little more closely, the CNS is made up again of your brain and your spinal cord. And it's primarily made up of interneurons and then the ends of sensory or motor neurons. And its job is information processing. Peripheral nervous system is basically everything outside the central nervous system. It's made up of your cranial nerves and spinal nerves. Afferents bring information to the CNS, and efferents bring things away from the CNS. When we look at the peripheral nervous system, we can look at the efferents, the motor ones, and look at the motor portion. All right, those go to skeletal muscle. They have voluntary movements, that's the body movements, and also involuntary movements that are reflexive. The autonomic goes primarily to smooth cardiac muscle and internal organs. It tends to be involuntary, and that's made up of two divisions. The parasympathetic, rest and digest, panda just hanging out in the tree, and sympathetic fight or flight with the panda getting ready to do kung fu. The parasympathetic, sometimes known as rest and digest or feed and breed, it tends to come out the cranial or sacral portion, and then the sympathetic comes out the thoracic and lumbar section. And again, you don't have to memorize these. Just kind of think of anything that gets you ready for fighting or running. That's going to probably be what the sympathetic nervous system is doing. And everything's getting you rest and recovery. That would be what the parasympathetic tends to do. Another way to look at the nervous system is divide it into the cellular portion of it. And there's two basic types of cells we see in the nervous system. Neurons, which are the functional cells, and then glial cells, which are the supportive cells. There are a number of different glial cells. You can see some of them here. Not all of them are here, but some of them. So you have, the ependymal cells, you have astrocytes, oligodendrocytes, and microglial cells in the CNS, and then you have Schwann cells. There's also some satellite cells not shown here in the peripheral nervous system. We're not going to worry about those except to look at the oligodendrocytes in the CNS and the Schwann cells, or sometimes known as neurolemmacytes, in the PNS, in the peripheral nervous system. And the reason why we care about these two types of cells is the oligodendrocytes responsible for making the myelin in the CNS, and the Schwann cells are responsible for producing the myelin in the PNS. If you look at the lighter blue area, those are the oligodendrocytes. So the CNS is the top here, right? It goes down right here. So there's the lighter blue ones with the myelin. And then the PNS, we have these white ones here. So we're going to kind of look at them a little bit. So first of all, what's myelin? Myelin are these concentric wrappings around the cell membrane. They do a couple things. One is they help to support the structure, but they also help to provide electrical insulation. What they do is they speed up signal transmission. And basically, doesn't quite work this way, but it's not a bad way to think about it, is there are areas where there are no myelin wrapping on the axons. So these are called the nodes, or the nodes of Reeve, and you see them both on this and this. So A is a drawing of a peripheral neuron, and that's the Schwann cells, because Schwann cells are found in the PNS. What's interesting about this is every Schwann cell is a distinct own Schwann cell. So this is a Schwann cell, there, 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 there. So you can actually see 5 Schwann cells in this picture. Now, in the CNS, the oligodendrocytes make the myelin. If you look at this picture, you have one oligodendrocyte, but notice it's got appendages to make all the myelin on these 2 axons. So instead of having a whole bunch of cells on the axon, it's one cell. That becomes important later on for our discussion. So Schwann cells have many, many, many cells on the axon because they're each independent, while oligodendrocytes have very few cells on the axon. And typically it's at least a 1-to-1 ratio, and sometimes a 1-to-5 ratio, meaning one oligodendrocyte may make 2, 3, 4, 5 different axons, while here you can have literally thousands of individual Schwann cells on an axon if it was long enough. So why this becomes interesting is years ago, Christopher Reeve, who played Superman, was in a horse riding accident and broke his neck and became a quadriplegic. About the same time in Florida was a little kid out swimming around by the name of Jesse Arbogast, and he was a shark attack victim. And basically what happened was he was out swimming around, and depending on what version of the story you want to listen to, the details are sketchy, but somehow he got his arm bitten off by a shark. His family was able to grab the shark, bring it up to shore, literally beat the shark to death. And then what they did is they took the boy and the severed arm to the hospital and they reattached it. And his arm works okay now. Not great, but okay. Christopher Reeve had the best medical care there is. And he really never made any progress in terms of his rehabilitation after a spinal cord injury. So the major difference was Christopher Reeve injured his central nervous system, while Jesse Arbogast had his peripheral nervous system injured. And so Christopher Reeve never got any better, and the CNS doesn't regenerate. And they believe what happens is at least partially due to the 1-to-1 or less relationship of the oligodendrocytes. When he has the injury to his neck, 100% of the oligodendrocytes got damaged. When Jesse Arbogast had his arm severed, there's many, many, many functional Schwann cells left that help the peripheral neurons regenerate and reconnect. And in fact, Schwann cells produce a chemical that allows them to basically find each other. So there is a difference in terms of their regeneration property. And so peripheral neurons regenerate while central neurons do not, at least as far as we can tell. So here's an example of a whole bunch of axons, a whole bunch of Schwann cells around an axon, right? So you see many, many, many of these. Looks like this. We'll talk about the function of those a little later. So when we see nerve damage, right, you still have the site of injury here, but you have functional, and usually there's hundreds and thousands of these, not just 5 or 6 here. that can help regenerate in terms of the peripheral neurons finding each other. And so that regrowth really helps in terms of recovery and things like that. So the peripheral neurons can regenerate their axons, but not their cell bodies. The central ones doesn't seem to be able to do either. And again, it has to do with this PNS Schwann cells. We've only damaged a small percentage of them. Here in the CNS, the oligodendrocytes, we've damaged basically all of them because of the ratio. So now we're going to focus in on neurons. And neurons are basically the functional cells of the nervous system. And we can look at neurons, and every neuron has 3 parts to it. And each part sort of serves as a microcosm of the entire nervous system. So the 3 basic parts of a neuron you'll see are dendrites, and those are the areas that receive input. The cell body, that's the integration and processing. And then the axon, that's the portion that transmits the output. So when we end the neuron, we end at a synapse. That's the junction between the neuron ending and the target cell. Synapses, mostly in the human body, are chemical synapses. So look at the bottom left here, where we, through exocytosis, release the neurotransmitter, diffuse across the synaptic cleft, binds to some sort of receptor on the end organ or target cell, and typically has some action. All right. Now, there's a couple different types of neurons, and we won't see them all here, but here's a couple. So we can divide the neurons up structurally, and we can divide neurons up functionally. All right, so I probably should reverse these two. Let's look at them structurally first, because that's what you're probably most familiar with from anatomy. So there are 3 basic types of neurons we can find. We'll start with the multipolar, because those are the ones you're most familiar with. So when neurons have the term polar, it really refers to how many appendages they have. So a multipolar neuron is going to always have this. Okay? It's going to have a single cell body. It's going to have a single axon exiting that cell body, although it can branch later on. Okay, so 1 axon, 1 cell body, but it's going to have many dendrites. Okay? And so that's where the multipolar comes from. It's got many appendages, and really anything more than 2 is considered a multipolar neuron. Okay? Now, we also see multipolar neurons here. So here's 1 axon, 1 cell body, and look at all these dendrites. All right, this one's a little more confusing, and this one's a little more confusing. And the anaxonic, we're really not going to worry about there. So this is the classical multipolar. Here's a couple other types of multipolar. And then we're going to have what's called a bipolar. Bi means 2, right? So you have a cell body, and here you have the dendrites here. This goes to an axon. This comes from the cell body and axon, and then here's where the end organ will be here. That's called a bipolar because it's got 2 appendages. So remember, polar is appendages too. And this last one is sometimes called unipolar, but nowadays mostly called pseudo-unipolar. Pseudo means fake or false. So again, here are the dendrites. Here are, I guess here is the axon. And then here's the cell body off to the side. Okay. So it's called pseudo-unipolar because it looks physically like it's got 1 attachment, when actually it's got 2-way traffic in that attachment. So say it's not really that. So it's pseudo-unipolar because it looks like 1, but is really 2-way traffic. So those are the structural classifications of neurons. Those are probably what you learned in anatomy. And that's great, except we're in physiology, so we want to look at the functional ones. So coming over here, the functional ones, kind of see this. We'll go back to the other one in a second. So functionally, we can look at a couple things. One is okay, afferents. Okay, so those are sensory neurons. And sensory neurons, this is one type. Here's the dendrites. This is going to pick up whatever sensation it is. It's going to transmit it down the axon, right, through the cell body, and then to the end organ. So the afferents, or sensory neurons, bring information into the CNS. Now the CNS is the green. Here's the peripheral nervous system, the PNS in the blue here. So this is the dividing line right here, going up and down. So in the CNS, right, we have this neuron where it has a cell body. It's got a whole bunch of dendrites, and it's got an axon, and it goes to another neuron. This neuron is completely enclosed within the central nervous system. It begins and ends there. That's called an interneuron. So any neuron that starts and ends in the central nervous system without leaving it is called an interneuron. Now here is our efferent, cell body in the CNS, but the signal will come from the CNS in the efferent neuron or motor neuron, and it'll go to the end organ, which is typically a muscle gland, another neuron, or something like that. So let's look and kind of classify the function based on structure. Okay, so we'll start here. This is the multipolar neuron. All of our motor neurons, whether it's somatic, so any of our efferents, whether it's somatic or autonomic, is going to have this multipolar shape. In addition, all of our interneurons within the CNS that begin and end in the CNS that are there for processing are going to be multipolar. So not surprisingly, by far, not even close, the majority of our neurons are structurally multipolar, right? And the multipolar ones are the efferents and the interneurons. The other two are part of the sensory system. The bipolar neurons are for special senses, especially for smell and vision. As a matter of fact, if you're familiar with the microscopic structure of the eye itself, then on the retina, where the image is basically displayed for the eye, we have special sets of neurons, literally called bipolar neurons. As a matter of fact, you're probably familiar with the receptors for that, the rods and the cones. They're just modified bipolar neurons. So bipolars for special senses, especially smell and vision. So that leaves the pseudo-unipolar for what we call the somatic or general senses. So we see the pseudo-unipolar in playing that role. So there's relatively few of the pseudo-unipolar, and there's really, really few of the bipolar, and many, many, many of the multipolar. So we'll pick up with the next video about how a synapse works when the neuron connects to another end organ. All right, so we'll spend some time in detail looking at this synapse.